Assalamu alaikum. In previous lecture we have discussed the origin and evolution of bacteria. Although bacterial fossils exist such as stromatolites, their lack of distinctive morphology prevents them from being used to examine the history of bacterial evolution or to date the time of origin of a particular bacterial species. However, gene sequences can be used to reconstruct the bacterial phylogeny, and these studies indicate that bacteria diverged first from the Archaea eukaryotic lineage. The most recent common ancestor of bacteria and Archaea was probably a hyperthermophile that lived about 2.5 billion to 3.5 billion years ago. The earliest life on the land may have been bacteria, some 3.22 billion years ago. Bacteria were also involved in the second great evolutionary divergence, that of Archaea and eukaryotes. Here, eukaryotes resulted from the entering of ancient bacteria into endosymbiotic associations with the ancestor of eukaryotic cells, which were themselves possibly related to the Archaea. This involved the engulfment by proto-eukaryotic cells of alpha proteobacterial symbionts to form either mitochondria or hydrogenosomes, which are still found in all known eukarya, sometimes in highly reduced form in ancient amitochondrial protozoa. Later, some eukaryotes that already contain mitochondria also engulfed cyanobacteria-like organisms, leading to the formation of chloroplast in algae and plants. This is known as primary endosymbiosis. If we discuss the habitat of bacteria, bacteria are ubiquitous, living in every possible habitat on the planet, including soil, underwater, deep in Earth's crust, and even such extreme environment as acidic hot springs and radioactive waste. There are approximately 2 multiplied by 10 raised to 30 bacteria on Earth, forming a biomass that is only exceeded by plants. They are abundant in lakes and oceans, in Arctic ice and geothermal springs, where they provide the nutrients needed to sustain life by converting the dissolved compounds such as hydrogen sulfide and methane to energy. They live on and in plants and animals. Most do not cause diseases, are beneficial to their environments, and are essential for life. The soil is a rich source of bacteria, and a few grams contain around 1,000 billions of them. They are all essential to soil ecology, breaking down toxic waste and recycling the nutrients. They are even found in the atmosphere, and 1 cubic meter of air holds around 100 million bacterial cells. The oceans and seas harbor around 3 multiplied by 10 raised to 26 bacteria, which provide up to 50% of oxygen humans breathe. Only around 2% of bacterial species have been fully studied. Extremophile bacteria, their habitat is cold -15 degrees in Antarctica. Species Cryptoendolithis, hot habitat 70 to 100 degree geysers, radiations 5 million rad, saline 47% salt, Dead Sea, Great Salt Lake, acid pH 3, alkaline pH 12.8, space 6 years on a NASA satellite, 3.2 kilometer underground, high pressure, Mariana Trench, 1200 atmosphere, high pressure. So bacteria can survive in these severe conditions. If we discuss the morphology of bacteria, then bacteria display a wide diversity of shapes and sizes. Bacterial cells are about 1/10 the size of eukaryotic cells and are typically 0.5 to 5.0 micrometers in length. However, a few species are visible to the unaided eye. For example, Theomargarita nambiensis is up to half a millimeter long. U. eplopsicum fischelsoni reaches 0.7 millimeter, and Theomargarita magnifica can reach even 2 centimeters in length, which is 50 times larger than other known bacteria. Among the smallest bacteria are members of the genus Mycoplasma, which measures only 0.3 micrometers, which is as small as the largest viruses. Some bacteria may be even smaller, but these ultra microbacteria are not well studied. If we discuss the shape, then we have to move in our another lecture. Thank you very much.